Good morning, everyone. We plan on finishing the microbial growth today. We should have plenty of time for that. As a kind of break, we're gonna see what's in the incubator right there with the gram-positives. It's gonna be pretty interesting, actually. Review the quiz. If we have time left, I will do the review of the study guide. Don't worry if we don't have time left, so if we finish tight, I'm gonna be in the whereabouts tomorrow. Whereabouts, I mean Lakeland whereabouts. So I'm gonna stop. You don't have to come, don't worry. I'm gonna stop by and do the recording of the study guide review. Okay? It will be on the YouTube so you can watch it over the weekend. That's what I do if we just don't have time. So the exam, plan on it for Monday. Okay? Sounds good? Good. So we talked yesterday, we finished our discussion with the history sort of travel, going back and finding out about scientists who made the greatest contributions to the field of antibiotic discovery. However, you appreciate, I hope, that it's more than just Alexander Fleming and Zellman Waxman who contributed to a current knowledge about antibiotics. So speaking of the clinical consideration, when you pick antibiotic to treat a certain disease, there are several things that you have to take into account. First and foremost, probably, whether the antibiotic is bactericidal or it is bacteriostatic. Okay? Bactericidal, of course, it's better. It completely destroys the microbe. However, it is not often possible. Some antibiotics are rather bacteriostatic, they just stop the replication rather than killing the microbes. Now, the spectrum of action. Obviously, antibiotics can be broad spectrum and narrow spectrum, and each of those classes has its own advantages. What is the advantage of broad spectrum antibiotic? What do you think? Can treat more, so if you don't know what the patient has, you can throw in broad spectrum antibiotic. What is the disadvantage of broad spectrum? Not necessarily. It can be very strong.
You're giving such a broad spectrum, you might develop antibiotic resistance to that because you're not specifically targeting to that bacteria or that microbe.
Exactly. You affect not only the microbe of interest, you're essentially treating all the microbes that the patient has, including the good ones, whether they are in the gut or in the vaginal mucosa, oral mucosa, So that's the problem with the broad spectrum. And that can be ameliorated by the narrow spectrum antibiotics, which are targeted specifically, suited specifically to a certain microbe. For instance, in many cases, when you get to the doctor with the complaints about strep throat or other types of respiratory infection, the major causative agent, strep throat, is Streptococcus pyogenes, which is the gram-positive microbe. So it's likely that doctor will prescribe something that targets specifically gram-positives, things like penicillins, for instance. Okay, well, I'm not saying penicillin per se, but it can be penicillin, it can be amoxicillin. Okay. So broad spectrum antibiotics essentially may lead to the development of superinfection, as it is illustrated at the bottom. When broad spectrum antibiotics eliminate all the non-resistant microbes, resistant ones will remain, and if they are pathogenic, maybe they are opportunistic pathogens, they may cause infection. Now, Have you heard about Clostridium difficile? I believe you did. You have heard about Clostridium difficile. Many people are asymptomatic carriers of Clostridium difficile. They have it in their guts. They do not develop any symptoms. However, when they get treated for various types of infections, it changes the balance of species in the gut. Clostridium difficile starts to outgrow other species, and when it proliferates, people develop C. diff, associated colitis. That makes sense? Route of administration. That is an important point of consideration. The most convenient, of course, is oral. And that's how we take antibiotics, right? But the problem is that some antibiotics can be absorbed in the gastrointestinal tract and some cannot, right? And oral antibiotic, as you can see on this graph, its plasma concentration doesn't elevate as high as it does for IV antibiotics. Okay, so for non-acute syndrome, non-acute disease, your strep throat, for example, okay, you find you can get away with oral antibiotic because your life is not in immediate threat. However, when patient is admitted to the, emergency room with the acute case of tularemia, which is a life-threatening condition, and respiratory tularemia can be lethal in 48 hours. In this case, there's no time to wait for that elevation in the plasma levels of antibiotics. So IV antibiotics should be prescribed. Does that make sense? In other cases, for instance, another great point of consideration, what type of infection, where is the infection? If it's in the accessible tissues, say heart, okay, then you may actually get away with a lower plasma levels of antibiotic. But if it is in the bones, for instance, like osteomyelitis, in case of severe acute osteomyelitis, patients have to get a catheter installed, a catheter in the subclavian vein, because they have to receive IV antibiotics for months, because the microbe that's in the bones is really hard to reach. Okay. Synergistic interactions. We're going to see that that's going to be exemplified by antibiotics belonging to the class of metabolite inhibitors. Trimethoprim and sulfamethoxazole, when taken individually, are bacteriostatic. When you combine them, the combination of them, that is called Bactrim, is bactericidal. Does that make sense? Now, where do we take them? We can synthesize them, like chloramphenicol, synthetic antibiotic, okay, or we can find them, or fluoroquinolones, or we can find them in nature, like penicillin. We did neomycin, actinomycin. We found those antibiotics in bacteria or fungi, and actually soil and the microbes that are in the environment serve as the great source of new antimicrobial drugs. And my question for you is, why? Why those microbes that are outside, how come that they have drugs against microbes?
At some point, you had to develop that
For what?
for their own growth. Like it was probably
To do what? You're in the right direction. Just to do what?
some competition.
Yep, exactly. It's the chemical warfare that they use against other microbes. Exactly. Now, one more question. Hypothetically, if you have two drugs, one is natural drug, okay? the one that you got from some fungus. And another one is synthetic. They are both just, natural is just isolated, just recently, just, I don't know, 2 days ago. And synthetic is synthesized 2 days ago. To which one the microbes are likely to have resistance? To natural one or to synthetic one? Exactly, natural. Can you explain why? Exactly, because they've seen it before. Yes, a lot of microbes have intrinsic resistance. Because if you have one microbe that produces a chemical and another microbe, I'm talking about species, that is exposed to that chemical, that second microbe, as the protective mechanism, develops the resistance. Does that make sense? So it's resistance, antibiotic resistance, is not only driven by us using antibiotics. It is a normal thing in the environmental microbes. Does that make sense to you? And due to the horizontal gene transfer that we discussed, due to the conjugation, due to the transduction, due to the transformation, antibiotic resistance is migrating from one microbe to another. We will talk about what contributes the most, whether it's us or the nature. We'll talk about it, okay? But so far, we just have to keep it in mind that it can happen. Now, this is the summary, the mechanisms that various antibiotics act through to prevent the bacterial growth. Some antibiotics, very successful antibiotics, target cell wall. Among them are different types of penicillins, cephalosporins, drugs like ceftriaxone, vancomycin, and bacitracin. Some target plasma membrane, like polymyxin B that you can find in the triple antibiotic ointment, Many antibiotics, and they are very efficient, target protein synthesis. They bind to ribosomes and do stuff. We have several groups of antibiotics that target DNA and RNA synthesis of nucleic acids. And we have several ones that inhibit enzymes involved in various metabolic pathways. Okay? I deliberately didn't go through every one because we're going to talk in details about every mechanism. Okay? And if you glanced through the slides, there should be a slide closer to the end, which is essentially the same, but without antibiotics. Does that make sense? So it has only, let me see, should be, yeah, this one. So see, you have targets, and you can, what you can do, I provided it for you as a study tool, okay? We're not going to, you can help me out in the end of today, okay, to fill out those targets, which antibiotics work in each. Does that make sense? Kind of study tool for you. Okay, let's go back to, I also made a table which breaks down the functions of those antibiotics. You can use it sort of a study guide. So imagine that you have a table which has only this column, okay? So you can put antibiotics with their relative mechanisms on the right. So you can fill up to this too. Or imagine you have a list of classes and you need to figure out the mode of action. You can just practice it, essentially preparing the sort of set of flashcards. I also have, I don't remember if it's 100% complete or if it's somewhat incomplete, set of flashcards online. The website is called cram.com. So you essentially go there. It's like Study Blue and all that kind of crap. And you look for my last name. I don't think you have to sign in to study. So you see my last, that's the author. Okay, you click on the author. I hope it'll show. Yeah. So I have a little bit antimicrobials, classes and mechanisms. It's probably not complete. If you like it, you can use it. Don't take it as the ultimate perfect thing and, you know, whatever. Okay? Does that make sense? It covers more than you need for the exam. Let me put it this way. It covers more than you need. But it may help you, and you can put the Cram app on your phone. And yes, I need to make this disclaimer. I do not get a penny from that company. I just find this website convenient, that's all. Okay, so there's no monetary interest. If you want to export, I don't know if you can, but if you want to kind of export it to Study Blue or any other online flashcard service that you use, more than welcome. It's public. Okay. Does that make sense? Oh, crap. It's a lot of, you're going to hear some voice on the recording. There was something online, some sort of an advertisement. So sorry about that. Anyway, let's talk about each individual classes. So first and foremost, it's the inhibition of cell wall. Almost all bacteria, except for a few, except for molecules, things like mycoplasma and ureaplasma, all other microbes have cell wall. Correct? So if you sum and think about this, they have not only, so first of all, how do bacterial cells divide? Binary fission. So before they divide, what happens to the mother cell? Replicates DNA. What happens to the size of the mother cell? It increases, so it has to make more, right? It has to make more of everything, including cell wall. If you can inhibit somehow the synthesis of that cell wall, you're going to inhibit essentially replication of microbes. And if cell wall is not built properly, for instance, cells actually can die. Not only they may not replicate, they will die eventually. Does that make sense? So there are three major classes of drugs that inhibit cell wall synthesis. One is beta-lactam antibiotics. There are several subclasses in that group. Okay? What they do, they inhibit the synthesis of peptidoglycan. They inhibit the formation of peptide links between the carbohydrate chains in the cell wall. Does that make sense to you? So essentially, instead of forming the mesh, as we discussed when we talked about cell wall, you now have disconnected chains of carbohydrates. So it decreases the rigidity of the cell wall and decreases it quite considerably. Okay? So these drugs that contain so-called beta-lactam ring, you can see beta-lactam ring on this picture. I circle it with a green marker now. So drugs that have beta-lactam ring, they bind to the enzyme, inhibit the enzyme called transpeptidase, also called penicillin binding protein, okay, and inhibit transpeptidase function. Does that make sense? So these drugs are bactericidal because they, when structure of the cell wall is affected in this way, cell wall becomes weak and cell lyses. Cell pretty much explodes. That makes sense to you? Okay. Now, of course, microbes could not develop the resistance, and a lot of microbes now have the enzyme or various types of enzyme called beta-lactamase. Beta-lactamase destroys the beta-lactam ring. Does that make sense? So if we will try to line out the mechanism of action and mechanism of resistance, it would look this way. I'm going to write and draw over this slide for a minute, right? So essentially, what is inhibited? Synthesis of what? Peptide links, right? Which enzyme facilitates it? Which enzyme? Transpeptidase. We're going to call it PBP, penicillin binding protein, just for, yeah, it's a synonym. Just called penicillin binding protein, unfortunately, because it binds penicillin. That's it. Okay? So PBP facilitates it. Okay? So what does penicillin or any other beta-lactam does? It inhibits PBP, so no links are formed. Does that make sense? So these guys inhibit PBP. Okay? In response, microbes have the enzyme called beta-lactamase, which does what? Destroys the beta-lactam rings. Essentially, killing the function of all these drugs. Now, the drug known as clavulanic acid allows physicians to get around that type of resistance. What do you think it does? I'm going to give you a hint. Think about enzyme inhibition. Clavulanic acid inhibits something. The answer to that something is right there. Clavulanic acid inhibits something, allowing us to use beta-lactams again. Exactly. Clavulanic acid is the beta-lactamase inhibitor. Okay? Does that make sense? When clavulanic acid inhibits beta-lactamase, beta-lactamase cannot break down beta-lactam rings, and beta-lactams are efficient again. Does that make sense to you? Clavulanic acid? It's a drug. I don't know whether I think they isolated it. It's natural antibiotic. I mean, it's not antibiotic per se. It kind of makes beta-lactams effective again. Often you can see the prescriptions for beta-lactams. The patient may be prescribed amoxicillin plus clavulonate, two things together, because amoxicillin itself may not be effective. Does that make sense? Okay. Now, one thing that I want to demonstrate here is so you may wonder, what's the difference between different beta-lactams? The difference is in the side chains. So you can see that this is the structure of penicillin. This is cephalosporin, so these parts are different. This is monobactam, and this is carbapenem. Okay, so these parts of the molecule are all different. Does that make sense? So it's side chains that change from one beta-lactam to another. Does it make sense? Changing those side chains, you can see the same situation in this table. So this table refers to different types of penicillins. You can see that penicillins have this part that is common for all penicillins, and R part that's different. Those are different R's for different types of penicillins, starting from penicillin G and finishing with methicillin. Look at the difference of activity. They have different spectrum of activity. Does that make sense? They have different route of administration. And let me tell you, different microbes have different resistance to those. So when we change the side chains, side groups of antibiotics, what do we change as well? Huh? Be more specific. What type of function? Huh? What type of structure? But it leads to change in function. In which functions? Two things. Okay, which bacteria they inhibit, so spectrum, and also what else? The resistance. We can overcome resistance by changing, tweaking antibiotic structure. Does that make sense? That actually brings farm companies pretty good money, because in terms of research and development, it's not like designing a completely new drug. Does that make sense? They tweak it a little bit and they market it, and they have to market it as a new drug. Okay. I never asked you, by the way, anyone knows, what is the cost to develop a completely new drug from scratch? Anyone knows? From like, I have an idea, let's go, to the moment we got it on the market, with all the clinical studies, research and development and whatnot. Regulatory fees. Huh? Say again? You pessimistic? A little less, but not much less. 1.5 to 2. Yes, for a drug. Bless you. So that's why, you know, you look at the prices of some drugs, and the thing is, when company patents the drug, to the moment, it's pretty much a cycle. But think about this: they spend so much money on the drug, they patent it, but they don't patent it when it goes on the market. They patent it while it doesn't even hit clinical trials, and clocks start to tick. So at the moment it hits the market, they have certain amount of time till the moment they have to open up the patent. That's when you get generic drugs. And they charge a living hell for each pill to compensate for their costs. Does that make sense? So that's the mechanism pharmaceutical market works. Now, those were beta-lactams. We have a couple more cell wall inhibitors. Vancomycin, which actually prevents the process known as transglycosylation. Essentially, vancomycin prevents the synthesis of these chains, the carbohydrate chains in the cell wall. And bacitracin, it's the natural antibiotic isolated from one of the bacillus species. It prevents the transport of molecules that serve as precursors for glycans via the cell membrane. So it inhibits the transport. So you cannot deliver the building blocks to the cell wall. Does that make sense? So this is a little bit, the illustration on the bottom shows you, what happens to the cell when there is no proper cell wall synthesis. The cell wall is weakened in cell lysis. Now this table shows you some of the drugs. You do not have to know the brand names. It's not the pharmacology class. And I will focus on, if generally you don't need to know the spectrum. If you do need, I will tell you the spectrum and I will tell you, remember that. Not only know, I want you to understand why this spectrum. So drugs like penicillins, okay, they are most effective against gram-positive microbes. Why? Not so much against gram-negatives. What's the difference? They have thicker cell wall, yes. They have thicker cell wall, so you inhibit the cell wall synthesis. And it's right there, okay? For gram-negatives, you have to get through the outer membrane, cell wall, inner membrane. It's just more troublesome, okay? Cephalosporins, you probably have heard those names. Things like ceftriaxone, cefdinir, okay? Those drugs have broad range. Okay, they're really good. Also, well, they all semi-synthetic. By the way, when we say semi-synthetic antibiotic, it means that we started off, with a natural drug, the drug that we isolated from some kind of a microorganism outside, and then we started to tweak it. Cephalosporins actually represent the great case for the arms race between humans and microbes. When microbes gained resistance to previous generation, humans developed the next generation. They gained resistance to this generation next. So now we are the 5th generation of cephalosporins. We keep tweaking. Okay. Monobactams. It's monobactam. You have to. This sort of a chemical class of compounds. Only one of them showed a promise. It's astrionam, semi-synthetic drug, and the most advanced beta-lactam antibiotics by now are carbapenems. Okay, they have probably the broadest range. They are effective against positive, gram-negative, and beta-lactamase carrying into, so resistant microbes. Does that make sense? Vancomycin, gram-positives, okay. Bacitracin, it's a broad spectrum. So, take-home message, what you have to know. Mechanisms at the level of beta-lactams inhibit formation of peptide links between the carbohydrate chains. Vancomycin inhibits the formation of carbohydrate chains per se. Base tracing inhibits the transport of building blocks of the cell wall from inside the cell to the cell wall. Is that clear? Is that clear? Range, the only thing that I can ask is why penicillins are so specific for treatment of gram-positives. Because they specifically target the cell wall, they cannot be effectively transported in the cell. Got it? So they specifically inhibit the cell wall, and gram-positives have thicker cell walls, so they're more sensitive to penicillins. Okay? That's the only question about the range that I'm going to ask, if I will, regarding these guys. Any questions before we move on to the next class? We're good? This inhibition of protein synthesis has become one of the major research targets in the quest for new promising antibiotics. The reason for that is, what do you think? What is the bacterial ribosomes and eukaryotic ribosomes are what? The same? Different. They are structurally different. And if things are structurally different, you can make specific inhibitors of one, but not another. Does that make sense, that statement? It's a reasonable statement, right? So, the great advantage of these drugs is their high specific toxicity. We didn't discuss it, let's talk about this now. What is specific toxicity? Or selective toxicity? Those are synonyms. Specific or selective toxicity. Come on, give me your versions. What do you think it means? If drug has high selective or high specific toxicity. Good. So it kills one organism but not others. Does that make sense? So these drugs, since they are targeted, they tweaked to interact with bacterial ribosome, they have high selective toxicity. By the way, on the same note, what do you think about the previous ones, the inhibitors of the cell wall synthesis? Do they have high selective toxicity? Why? Explain your answer.
Because, well, they're more broad spectrum.
I'm talking not about between the microbes. I'm talking about bacteria versus humans. Does that now, considering this, do they have high selective toxicity? Why? What?
Well, they harm the bacteria, not us.
Why they do not harm us? What do we not have? There you go. Does that make sense? They inhibit something that we don't have. So yeah, we can have allergy to penicillin. That's fine. Okay, but we don't have cell wall. There's nothing to inhibit. They're not going to hurt human cells in the same way they hurt bacterial, right? So these guys have high selective toxicity as well, the protein synthesis inhibitors. Most of them either come from natural sources or used to come from natural sources, came from natural sources and later got tweaked. So synthetic or semi-synthetic, with the exception of oxazolidinones, which are synthetic drugs. Okay. So this picture shows you the main classes and what they inhibit. So basically, you can see that this three classes, chloramphenicol, macrolides, and lincosamides, bind to the 50S subunit and somehow prevent the formation of the bond between the amino acid, incoming amino acid, and the growing polypeptide chain. Aminoglycosides and tetracyclines bind to the 30S subunit and either leads to the formation of malfunctioning protein, like aminoglycosides, or prevent the tRNA binding, like tetracyclines. Lincosamides, oxazolidinones, altogether, they inhibit formation of initiation complex. We're going to talk about each class of drugs separately. In the black boxes, you can see here, those, it's entirely FYI. I'm not going to ask about side effects of antibiotics, but I just show you the side effects. Okay? Some most notable. For instance, for chloramphenicol, very common side effect is so-called aplastic anemia, when bone marrow is damaged to the level that it stops producing red blood cells. For aminoglycosides, just all through, so kidneys, nervous system, and ototoxic. It decreases leads to the hearing loss. For tetracyclines, very well known side effects is phototoxicity. So they become toxic when people are exposed to the sunlight. Okay. So this is the overview of drugs, major classes of drugs that inhibit protein synthesis. So first we're going to talk about the inhibitors of 30S subunit of the bacterial ribosome. Aminoglycosides, such as this drug, streptomycin, gentamicin, neomycin, kanamycin, they bind to the bacterial ribosome. You can see it on this upper left corner, the pink triangles, they bind to the 30S ribosome. And disrupt the structure of the elongation complex. It doesn't prevent protein from being formed, okay? But the problem is that proteins that are synthesized as the result are bad. They're faulty. When these faulty proteins, after leaving the ribosome, get into the cell and start to, well, proteins doesn't mutate, but it doesn't work right. I mean, if you stick the wrong part into the engine's gonna, stall or explode, something like that. So that's what happens to the cells. They die because proteins don't do their job. They're faulty. Okay, that's aminoglycosides. Tetracyclines inhibits the binding of tRNA to the A site of the ribosome. And to be honest, I bet you've heard about these drugs like tetracycline or doxycycline. These are the broadest spectrum drugs that we have. They may be, carbapenems may be comparable, okay? But tetracyclines, for a very long time, that's the drug of choice in treatment of unknown microbe, okay? And I'm just waiting when we get to the bed bugs, and I'm going to tell you clinical stories. Okay, so that's U2 30S subunit inhibitors, 50S subunit inhibitors. Macrolides, lincosamides, and chloramphenicol. So you can see that chloramphenicol prevents the formation of the peptide bond directly. Okay, erythromycin, one of the macrolides, the one that we tested recently, it prevents the ribosome from moving forward. Okay, lincomycin and clindamycin, as far as I remember, it doesn't let the tRNA to leave. And oxazolidinone, this drug, okay, it actually prevents the formation of initiation complex. See that here? So to which level you have to understand the inhibitors of the protein synthesis. You have to understand what they inhibit. Okay, 30S subunit or 50S subunit. For chloramphenicol, macrolides, and lincosamides, all you have to know is inhibition of the 50S subunit. For tetracyclines and aminoglycosides, all you have to know is the inhibition of the 30S subunit function. For linezolid, it prevents the formation of initiation complex. Am I clear on this? Related things that you also have to know: the reason why these drugs have high selective toxicity, which is discussed because of the structural differences between pro and eukaryotic ribosomes. Okay, and what else did we mention? Oh, the spectrum of action. Okay, what is the reason why tetracycline, for instance, have a broad spectrum? Because similarities, all microbes, all bacteria have very similar structures of ribosome. Okay, so tetracycline is capable of inhibiting that 30S subunit in all microbes. Does that make sense? Okay. Okay, membrane disruption. This is quite an interesting story. This antibiotics, polymyxins, and lipopeptides, they disrupt the structure of the bacterial membrane, not the wall, membrane. Polymyxins can bind to the lipopolysaccharides of the outer membrane of the gram-negatives, and they also can insert themselves into the inner membrane of the gram-negatives, essentially disrupting both and leading to the cell death. Okay, so they disrupt the structure of the membrane. They have pretty narrow range, targeting mostly gram-negatives. Okay, so far, keeping up with me. So disruption of the membrane. Daptomycin is the drug belonging to the class of lipopeptides. Its mode of action is quite fascinating. It inserts itself in the membrane, and eventually several molecules of daptomycin form a pore. Okay? And that pore now allows the flow of ions across the bacterial membrane, eventually leading to ionic imbalances, osmotic water flow, and cell lysis. So it's kind of piercing the membrane. Now, what are they used for? Polymyxin B is used for topical treatments. You can find it in the antibacterial ointments. Polymyxin E, or colistin, is used to treat severe bacterial infections. Okay, things like Clostridium difficile. Okay, severe infection, frequently antibiotic resistant. You need some sort of a nuclear bomb to just eliminate everything. Bowel decontamination. Something that can be used in immunocompromised patients, in which immune system cannot complement the action of the drugs. If it is used IV, it is used for treatment of systemic infection by, what's MDR, what do you think? multiple drug resistant microbes, okay? Multiple drug resistant. So think about this. Infections in immunocompromised people. Infections with multiple drug resistant microbes. Are those severe infections? Yes. So colistin is used to treat very severe infections. Okay? Polymyxin B, you can see that polymyxin B is not used internally. So these guys are not used to treat things like, I don't know, strep throat. Okay? Why? Same goes for daptomycin. Either skin infection, or if patient is bacteremic, it should be something extremely serious. Okay, like MRSA. Why it is used to treat such severe infections only? Why it is not used to treat?
Looks like they're very targeted.
Against what?
Appears almost like more resistant type of microbes.
What is the mechanism of action? What do they do?
This one breaks down the membranes versus.
What's the problem with that? Do you see the problem with antibiotics that disrupt the membranes? Oh yes, big time. These antibiotics are extremely toxic. Colistin leads to severe kidney problems. Colistin is often a last resort antibiotic in treatment of multiple drug resistant infections. Can be tuberculosis, multiple drug resistant tuberculosis. It can be multiple drug resistant gram negatives like Enterobacteriaceae, I don't know, E. coli, Enterobacter, and things like that, Klebsiella. It's really a last resort. Does that make sense? And a year ago, really horrible thing was reported. Plasmid-mediated colistin resistance. Yeah, which is bad. Think about this. It's not a chromosomal gene. It's a plasmid gene. Which means it can be transferred like this from one microbe to another. So I want you to, on these guys, what you have to understand, so the mechanism, of course, inhibition, disruption of the membranes for colistin, polymyxin B, and daptomycin. And the reason why these antibiotics are used for topical treatments mostly is because they have high toxicity for humans, high level of side effects. Does that make sense? So they have high level of side effects because of the mechanism that they have. That makes sense. Got it? So regularly we can use them for skin infections because it's not systemic use, it's topical use. Any questions? Replication, it should be actually not only DNA, like nucleic acid synthesis, honestly. Should have corrected the title. So, two drugs. One inhibits the DNA replication, one inhibits the RNA synthesis. Rifamycin, and the drug that belongs to the class of rifamycins is rifampin, inhibits the synthesis of RNA during the process of transcription. It binds to, it is a competitive inhibitor of RNA polymerase. It binds to the active site of RNA polymerase. Kind of loosely illustrated here. Okay? So you can see it binds to RNA polymerase so that RNA cannot leave the active site. Does that make sense? There is no transcription, cells cannot grow. The great thing about rifampin and as a drug, it's quite effective in the therapy of Mycobacterium tuberculosis. That makes sense? I'm gonna, why Mycobacterium tuberculosis? Why we focus so much on Mycobacterium tuberculosis? Why we say, oh, gram-positive, gram-negatives, and then separately Mycobacterium tuberculosis? Is it different? Is it different from gram-positives and gram-negatives? Yes. In what sense? It has mycolic acid on the outside. Okay? It technically has a gram-positive cell wall, but it is additionally covered by mycolic acid. It's like a bacteria covered with a layer of wax. For antibiotics, it's really hard to get in. Another problem with mycobacteria, they're slowly replicating bugs. So if you think about that, it's kind of a little digression, but in the same way, think about this. Which microbes are easier to kill? Let's even which cells can you kill? Cells that divide, grow and divide, or cells that are quiescent. Quiescent means nothing really happens. Cellular processes are stalled. Cells are technically alive, they just don't do anything. Which ones are easier to kill? Exactly, the living cells, the ones that replicate, the ones that reproduce. Because you can inhibit cellular processes. When nothing happens, there's nothing to inhibit. Does that make sense? That's Mycobacterium replicates very slowly. And it's really hard to kill slowly replicating microbes. Because you may have drug levels in the blood, going up and then going down. And during that the increased level of a drug, the microbe will just not replicate. It just doesn't hit in the right time. Does that make sense?
Is that why TB treatment takes so long?
Yes, absolutely. There are other reasons. It's intracellular pathogen. It gets inside of the macrophages, so it's hard to get there. Okay? People with some estimations, I'm kind of jumping completely different story. Just some estimations suggest that a third of the world population is infected with TB. They just don't get sick. A lot of people are carriers. Maybe not in the United States, but a lot of people are carriers. So that's one class. That's RNA polymerase inhibitor. The fluoroquinolones are inhibitors of DNA gyrase, topoisomerase 2. Remember we talked about the uncoiling of DNA? So DNA gyrase uncoils the DNA. If it doesn't uncoil, DNA replication stops. That makes sense? So you've heard about Cipro, broad spectrum antibiotics. A lot of those drugs, actually, if you would read about them, many of those, ciprofloxacin, vancomycin, tetracycline, amoxicillin, they are on the list of essential drugs, okay, that is, compiled and reviewed constantly by WHO, World Health Organization. So this is something sort of that every country, every clinic must have, okay? It's interesting also to see the prices of these drugs per pill and compare them between the US and Africa. It's much more expensive here because pharmaceutical companies, insurance companies will cover. You can't really sell anything for like $4 a pill in Africa. But, huh? Oh yeah, yeah. I bet there is a price difference. Yeah, but as well as prices for medical schools. I don't even want to touch it. 6-foot pole. So that was nucleic acid. what you have to know about nucleic acid synthesis inhibitors. What do they inhibit? So rifamycin inhibits RNA synthesis, fluoroquinolones inhibit DNA synthesis. Rifamycin is the competitive inhibitor of RNA polymerase. Okay. Sulfa drugs. That is a pretty interesting example of enzyme inhibitors. One of the key pathways in bacterial cells is the synthesis of tetrahydrofolic acid. Look on the right, so that pathway. It starts with a molecule called para-aminobenzoic acid, or PABA, which binds to the enzyme called dihydropteroate synthetase. So enzyme 1, okay, which produces dihydrofolic acid. Dihydrofolic acid is then converted in tetrahydrofolic acid by the dihydrofolate reductase, or we're going to call it enzyme 2. And eventually tetrahydrofolic acid is used as the substrate for synthesis of nucleotides, components of DNA. Correct? Makes sense? Now, there are two drugs that can inhibit either enzyme 1 or enzyme 2. One drug is type of drugs, sulfa drugs, sulfonamides. Remember we talked about the discovery by Domag and other German guys, the Prontosil? That was the first sulfonamide that was actually discovered. If you would look at the structure of typical sulfonamide and structure of para-aminobenzoic acid, you can see that those structures are fairly similar. Can you spot that? Okay, for some reason this thing doesn't work. Okay, can you spot that? You can do it with a mouse, but not with a... Right? So this one and this one is similar. Let's see. Okay, yeah. Okay. Now, sulfonamide. You can see it right here. Now, this sulfonamide inhibits the first enzyme. You don't have to know the names of the enzymes, okay? And prevents the pathway from going forward. Trimethoprim, right here, inhibits the second enzyme and also prevents the pathway from going forward, because trimethoprim has a structure fairly similar to the structure of dihydrofolic acid. So if those inhibitors are structurally similar, which site of the enzyme are going to bind? They're structurally similar to substrate, huh? Similar to the structure of the substrate. Going to bind active site. So these are both trimethoprim and sulfonamide are competitive inhibitors of the enzymes in the pathway of tetrahydrofolic acid synthesis. Does that make sense? Okay? Now, another example of synergism here is the fact that when you combine trimethoprim and sulfonamides in a drug that we know as Bactrim, together they become bactericidal, while individually they are bacteriostatic. Okay. Another drug that's not on the list, I'm going to mention that, the sulfur drug called dapsone. You don't have to write it down. I'm not going to ask it. Dapsone is an interesting drug. It's pretty unique. It used to treat leprosy. You have heard about leprosy, right? It's not that common in the United States anymore, okay? But it happens from time to time, especially in the southern states, because the main animal reservoir for leprosy is armadillos. I mean, it's really hard to catch one, but just, consider this. Now, other antimetabolites. Isoniazid is the drug that inhibits the synthesis of the mycolic acid. Okay? In which microbes are you going to find mycolic acid? Mycolic acid. Mycobacteria. Mycobacterium tuberculosis. Isoniazid is a drug very specifically used for treatment of Mycobacterium tuberculosis. Okay. Diryl quinolones. The drugs that Diryl quinolones are the drugs that interfere with the synthesis of the ATP. Okay. They inhibit some proteins of electron transport chain. Okay, we boned. Proteins of electron transport chain, and they're quite effective against mycobacteria. The problem is that they're pretty toxic because electron transport chain proteins, they're quite conservative. They're similar between mycobacteria, for instance, and humans. Does that make sense? Okay, so they have very high toxicity for humans and used for treatment of drug-resistant strains of mycobacterium. Take a message from this. Now, what they inhibit. You may get a question, what is the mechanism for isoniazid? What is the mechanism for diryl quinolone? So what is the mechanism for trimethoprim or sulfa drugs? Know that sulfa and trimethoprim are competitive inhibitors of, I'm not going to ask you the names of the enzymes, don't worry. Okay? Now, let's do this. This thing works again. Let's go over it. Let's name the inhibitors of the bacterial cell wall. I know how it works. Come on. Okay. Penicillins. What else? Cephalosporins. Try not to look at the original one. I know that's hard. How we call them all together? Start with this. Beta-lactams, okay? We have monobactam also, and carbapenems. Okay, so those are beta-lactams. What is the mechanism of resistance for them? Beta lactamase, there you go. Okay, good. What do they inhibit? Formation of what? No peptide links. Okay. What else? There are two more. The first letter is V, vancomycin. If it's not pep, what is cell wall made of? Peptidoglycan. So we covered peptido. Vanco inhibits glycan, synthesis of carbohydrate chains. Okay. Got it? And the last one is bacitracin. So no transport. So far, keeping up? Let's do plasma membrane. There are two of them, two classes. Polymyxins, which include B and colistin. Okay. And another one is daptomycin. Right? Polymyxins bind to LPS and disrupt the structure of the membrane. Daptomycin essentially forms the pores in the membrane. Inhibitors of DNA synthesis. Well, okay. Nucleic acid. What are those? There are two classes. Rifamycin and fluoroquinolones. Okay. Inhibitors of the protein synthesis. Ribosomes. What are they made of? 2 units. 30S. What are the inhibitors of 30S? Tetracyclines. Very good, Taylor. And aminoglycosides. Okay. Inhibitors of 50S. Macrolides like erythromycin. Lincosamides. Clindamycin. And chloramphenicol. And there's one more that inhibits actually the formation of initiation complex. Oxazolidinone. Well, it's a class, oxazolidinones. Okay. And inhibitors of metabolic pathways. So folic acid synthesis. Which ones? Sulfa and trimethoprim. Mycolic acid synthesis. So niazid. And another class, the, let's call it ATP synthesis inhibitors, diaryl quinolones. Okay. So my suggestion, print out that slide. Okay, and just do it by hand. You know why I ask? Why? My personal reason. That's my observation. When you do this, when you look at the picture, you make connections, memory connections, with the structures on the picture. So you can see that these guys, they inhibit DNA. Well, nucleic acid synthesis. You see that these guys, they target ribosomes. Does that make sense? So it improves your memory, because this is mostly, this is memorization. You can build some associative connections, but that's it. Okay, we're gonna talk about resistance later. Let's take a break.